Hello everyone, I welcome you all to the 6th lecture of this course, that is on DNA nanotechnology. In this lecture we are going to learn what is self assembly and what are the attractive features of self assembly and what drives this self assembly, and we are also going to learn what is DNA nanotechnology and applications of DNA nanotechnology, and we are also going to learn what is DNA origami and what are the drawbacks of DNA origami. So here the self assembly is a bottom up fabrication. Okay. So in my first lecture I told you what is top down approach and bottom up approach. Top down means from bulk to nanostructures, and bottom up approach means from atomic scale to nanoscale. Okay. So here we start with the small scale components and design the larger structures. So what is self assembly? Self assembly is a spontaneous organization of molecules or objects into well defined aggregates via non covalent interactions or forces. Okay. So here the building blocks is your molecules or any objects with the coded information for self assembly, and here the process is mix and shake and it will form the product. Okay. For example, here if you are using a DNA molecule, it have the coded information. So the DNA has A, it can forms bond with T, and G forms bond with C. Okay. So that means the molecule is already having the coded information. So when you put it in a solution, so the DNA easily forms bonds. Okay. So let us see a simple example to understand what is self assembly. So this you might have played in your childhood. Okay. So here the balls could be organized into triangles by an external agent or a player. Okay. Or what you can do is you can punch holes in the desired pattern and you can roll a stream of balls over the board. So here you can make a holes in this board, okay, and put the balls and shake it, so it will automatically go and assemble. So this is a simple example to understand what is self assembly. So let us see what is molecular self assembly. So one of the most important types of self assembly in nanoscience is molecular self assembly. So here the group of molecules spontaneously rearrange their positions in order to form the most stable structure. Okay. So which arrangement is most stable and why? So here the one which has the lowest interaction energy is the most stable. So usually the nature favors which requires low interaction energy and also high stable structure. Okay. So how the molecules move to form most stable structure? So at room temperatures the molecules constantly move around in space and change shapes due to heat or thermal energy in the sample. Okay. So if a collection of molecules is not in the most favorable configuration, that means not in the lowest energy form, so they will move towards most favorable configuration using thermal energy to move to the highly stable structure. So the nature always, as I told you earlier, it always favors the which requires low energy and highly stable structure. Okay. So the simple example is the tendency of water to flow from mountain side to valley. Okay. So what are the attractive features of self assembly? The self assembly proceeds spontaneously and it is often close to thermodynamic equilibrium. Okay. And again it rejects the defects and also it has a self healing capability, and self assembly is one of the few practical strategies for making the nanostructures. Okay. So let us see some other examples. Suppose you are having a molecules, two-ended molecules in the water. For example, this kind of molecules which have the polar head, that means water liking or hydrophilic molecule, and it is having non-polar trails, that is hydrophobic. So when you put this kind of molecules in water, what happens? All the hydrophilic heads will be facing the water and all the hydrophobic tails will away from the water. Okay. So you will get this kind of micellar structure. A simple example is if you put a drop of oil in a water, you will get a circular ring kind of structure, right. So the another example is we can make a nanoparticle. In this case they made a silver nanocubes, okay, and they coated with the hydrophobic ligand. So when you coat with hydrophobic ligand on only one side of this cube, so you will get this kind of assembly, and when you coat on the both the sides you will get this kind of line kind of structure, and when you coat it on the four sides, all the four sides, you will get this kind of assembly, and when you mix the four side coated nanocubes with the one side coated nanocubes, you will get this kind of assembly, and when you place like a six sided coated, you will get this kind of assembly. So, I will explain you in detail like. So, this is your nanocubes and this is coated with the hydrophobic ligand. Okay. So, it will join together, it forms this kind of structure. If you are coating these nanocubes on only one side, okay, and if you are coating these nanocubes on both the sides, it will form this kind of assembly. So, you can understand from this picture. So, if you are coating it on only one side, you will get this kind of assembly, and if you are coating on the both the sides, you will get this kind of assembly. So, why we need organization at the nanoscale? Okay. So, the synthesis of diverse nanoparticle is well developed. We have developed various methods to synthesize nanoparticles, but the challenging task is how to arrange these nanoparticles. Okay. Challenging part is their organization at the nanoscale. Okay. So, the multidimensional assembly of nanoparticles with controlled morphology is very important for realizing their novel applications. Okay. So, the organized or self-assembled nanostructures, it will show a remarkable collective properties which will be useful for engineering nano architectures. A simple example is, so if these all the alphabets are scattered, you do not get any meaning. So, these alphabets should be arranged in a proper way to get the meaning for the word nanoscale. So, similarly, if the nanoparticles are dispersed or scattered, you do not get any meaning. So, you have to arrange in a proper structure, okay. So, then only you can get various novel properties. For example, the physical properties and optical properties of the silver and magnetic materials is different, okay, when compared to the individual properties. And another thing is, it is a key to successful application of nanoparticles based devices, okay, and in this self assembly we will get the defect free ordered nanostructures. Okay. So, we can create periodic assembled nanostructures through different and novel and innovative procedures. So, which has a various application. By using self assembly, we can arrange these nanoparticles and we can make a various nanostructures which will have lot of application. Okay. And how do we can do it? We can do this nano organization by using top down approach like lithography, but in this lecture we are going to learn how we can use this bottom up approach for self assembly. Okay. Again, this self assembly is divided into two types, the static assembly and dynamic assembly. So, the static assembly is thermodynamic free energy minimum. Okay. So, once it is formed, it is stable, and dynamic assembly, it is kinetically formed, it is not necessarily thermodynamic minimum and not necessarily stable. Okay. So, what are the forces driving this self assembly? So, you can see that these are the forces of chemical bonding, for example, non-covalent interactions, ionic van der Waals and hydrogen, and other forces such as magnetic as well as hydrophilic or hydrophobic, and these are some of the forces which play a major role in driving this self assembly. And let us see what are the chemical interactions play a role in this self assembly. So, mainly the weak interactions play a major role in this self assembly. Okay. So, let us see example, hydrogen bond. It is a special type of attractive molecular interaction between electronegative atoms such as oxygen and a hydrogen atom that is directly bonded to another electronegative atom. And you know what is electrostatic or Coulomb attraction, that means, so it is forming between the positively or negatively charged atoms in a molecule, that is called as electrostatic attraction. The third one is metal ligand interaction. So, here the certain types of metals such as iron form a weak bonds with the specific atoms in the molecules, and the van der Waals interaction. So, here the weak attraction between the non-polar molecules such as those present in the automotive oils, and hydrophilic interactions, that means the tendency for charged groups in molecules to be attracted to water when used as a solvent. The last one is the hydrophobic interaction, that means the tendency of non-polar substance to aggregate in aqueous solution and exclude the water molecules. Okay. So, let us see what is DNA nanotechnology. Okay. So, DNA nanotechnology uses the bottom up, that is self assembly approach for making DNA based nanostructures. So, here the DNA nanotechnology uses unique molecular recognition properties of DNA, and it creates a self assembling branched DNA complex with useful properties. Okay. So, here DNA is used as a structural material. So, usually the DNA is a genetic material which carries the biological information, but here the DNA will be used as a structural material for making different nanostructures. So, that is called as DNA origami method. So, the DNA origami is the nanoscale folding of DNA to create arbitrary 2 or 3 dimensional shapes at the nanoscale. So, you know what is paper origami? So, using a paper we can make a different kind of structures. Okay. So, similarly you can use the DNA to make different kind of structures. So, this is a paper origami, you can use the paper and we can make different kind of shapes or structures, and if you are trying to mimic the similar kind of things using DNA, that is called as DNA origami. So, let us see some basics. Okay. So, the nucleic acid contains linear polymer of nucleotides. So, what is nucleotide, what is nucleoside? So, the DNA contains sugar, base and phosphate. So, the sugar plus base is nucleoside and the combination of sugar, base and phosphate is nucleotide. In case of DNA we will have 2 deoxy-D-ribose sugar, in case of RNA it contains D-ribose sugar. Okay. So, here the base is in simple you can ATGC, that is your adenine, thymine, guanine and cytosine. Okay. And we have to understand what is Chargaff rules, that is very important in this DNA nanotechnology. Okay. So, this A always form bond with T. Okay. You can see here this adenine always form bond with thymine using 2 hydrogen bonds, and other thing is the cytosine bonds with guanine with 3 hydrogen bonds. Okay. So, this is the Chargaff rules, and where A always bonds with T and G always pair with the C. So, this specific interaction between the complementary base pairs, that means like A how it forms bond with T and G forms with C. So, that makes the DNA as a attractive material, attractive construction material. So, using that property we can make different kind of nanostructures. Okay. So, for experimental synthesis of DNA nanostructure we need more amount of DNA. So, how we can synthesize the DNA? So, these oligonucleotides can be synthesized by DNA synthesizer and it can be purified by techniques like electrophoresis or chromatography, and it can be mixed together at the stoichiometric molar ratio in a near neutral buffer containing divalent cations. Okay. And if you want to denature the DNA, you can heat the DNA, that will break the hydrogen bonds and it will denature the DNA, and if you want to make it renature, it can be cooled so that that single stranded DNA combine and forms the double stranded DNA. Okay. And this is a very good example for the self assembly. So, let us see what is DNA hybridization. So, as I told you, this A always form bonds with T and G always forms bond with C. Okay. So, when you have the DNA like this and this is your complementary based on the other strand and it forms the hydrogen bond. So, when you apply this heat, what happens is this double stranded DNA became a single stranded DNA, and when you cool it again it forms the double stranded DNA. So, when you apply heat it will break the hydrogen bonds and it will become a single stranded DNA, and when you reduce the temperature and allow it to cool it will form a again double stranded DNA, that is called as renaturation. So, this is your denaturation and this is called renaturation. So, that is called as DNA hybridization. So, here you can hear simple example. So, you can use that color code. So, you can see here this green color DNA is binding to green color DNA and red color is binding to red color. So, it is like this sequence are complementary to the other sequence. So, that is why it is forming a double stranded DNA. So, when you apply the heat it can be become single stranded, and when you remove the temperature it can be like a double stranded DNA. So, using this we are going to make different kind of nanostructures. So, next thing is why the DNA is helical in nature. Okay. So, usually it can be like a ladder like structure. So, why it is forming this kind of helical structure? As I told you, the DNA has three main components: sugar, okay, phosphate, and base, that is a nitrogenous base ATGC. Okay. So, in this, these two are hydrophilic and this is hydrophobic, and inside the cell is full of water, right. So, what happens is, if we have a DNA like this, like a ladder structure, the water molecules can disturb the structure and this base has to be escape from the water, because this is hydrophobic. So, this base will be inside, okay, and your sugar and phosphate will be on the outside, because these are water loving, okay, and these base are hydrophobic, that is water repellent. Or so, after that also it can be like this structure, even the base can be inside. So, then also it can be like a DNA ladder like structure. So, why it has to attain like this kind of helical like structure? Because if we have this kind of space gap, again the water molecule can enter inside and it can disturb this structure. So, that is why it has to attain this kind of helical kind of structure to avoid the entry of water molecule. Okay. And another reason is the bonding angle between these A and T and G and C also plays a major role in giving this helical structure. Another simple analogy to understand why it is helical is, suppose your cloth is wet. So, what you do? You twist your cloth to remove the water, right. The same principle here also. So, the base is hydrophobic, so it has to be protected. So, it is forming a helical like structure. So, it is tightly coiled and not allowing the water to enter and disturb the structure. So, that is why the DNA is helical like structure, not a ladder like structure. And why we have to select DNA for making these nanostructures? Okay. So, because the DNA is the appropriate nanotechnology method for several reasons. The first one is it is relatively stable chemical and which exists in different forms, okay. And again as a polymer it can form a very long molecules and it has a well defined and repetitive structure. And again the rules for determining the structure are simple and well understood, okay. And within the molecule many atoms are available to form useful interaction and modification, and finally the DNA is again biocompatible material, okay. So, that is why we are using DNA as a structural material for constructing various nanostructures. So, we can use the DNA as a building material. So, here two important enzymes play a major role. The first one is restriction enzyme, okay. So, this enzyme is also called as molecular scissor. So, it can cut the DNA at specific site, okay, and another enzyme is ligase enzyme. So, it can join the two DNA fragments. So, using this self assembly principle, we can make a different kind of nanostructures and we can reach even 2-dimensional lattice. Here you can see here this is a DNA based tile, okay, and the tile will be arranged and form the lattice like structure. These are called as DNA tiles and DNA lattices. So, using this self assembly and complementary base pairing, we can make 1 dimension and we can also achieve this kind of higher dimension structures, and we can also make a DNA truncated octahedron structures, okay. And here it need about 8 bases must be paired for a double helix to be stable at room temperature, okay. And this DNA based 4-way crossover structures producing a rigid planar tile, okay, and the distance between the adjacent tile is 20 nanometer. And as I told you in the previous lecture, for biological molecule like DNA or protein, we have to use the AFM for understanding the structure. So, this is a AFM picture and you can see here how it forms a beautiful lattice structure. Let us see how to make 100 nanometers smiley faces using this DNA, okay. So, here we can use a viral genome which is of 7249 bases long, okay, and this viral genome will be made into this kind of smiley structures and using a small strands. That means we are going to use like more than 250 short strands and we are going to make this kind of smiley structure. So, these short strands are called as also called as staple strands. So, for example, if you want to staple the bunch of papers, you need a staple, right. Similarly, the DNA can be joined together using these short fragments of DNA, that is called as staple strand. So, if you want to make this smiley, the first step is you should have a plan how to make this smiley structure, okay. So, here you can see here that DNA is 2 nanometer diameter and the length is 3.6 nanometer for each helix. So, this is your first step to understand how many staple strands you need to connect the genome to make the smiley shape. So, once you made the plan, so then the second step is, so based on the plan you have to draw the structure, what are the what kind of smiley you are going to make, and then you will make the DNA short fragment sequences. So, that will bind to this and you can add some enzymes which will make the complete DNA sequence and finally you will get the complete structure. So, here the long template strand is annealed with a number of short strands, okay. So, that either form a cross links at fixed points or loops and to fill the regions to form the double helices. So, we will get the 100 nanometer smiley face using this DNA. So, similarly we can make a different kind of structures, okay, using this DNA as a construction material, and these are the free softwares available to design various nanostructures using the DNA as a construction material. So, let us see the applications of DNA nanotechnology. The first one is smart therapeutics and the second one is drug delivery. So, in the smart therapeutics it will release the drug when there is a particular signal is available, and we can also use it as a kind of nano carrier for drug delivery, and we can use this DNA for imaging application for diagnosing various diseases, and also we can use it for various cell biology applications. Let us see one by one. The first one I told you, like it can act like a DNA cage and it can carry any kind of drug. So, we can load with any kind of anti-cancer drug or any drug and which can be transferred to the cell and it can kill the cells, okay. And we can also make a DNA based box, okay. So, this box can be loaded with anti-cancer drug or any therapeutical molecule and it has a lock. This lock is also made by nucleic acid only. So, when you have the key of oligonucleotides, this will open this box and it will release the drugs. So, we can advance this DNA based box, okay. So, this hollow DNA based box can be assembled with the lid, okay, and it could be opened by a strand displacement with the specific oligonucleotide key. So, this box can be opened with the oligonucleotide key. So, if you have a DNA sequence that will open this box and you can make this kind of box with 2 different kind of anti-cancer drugs or one box with the imaging agent. So, that is called as theranostic nanoparticle. That means, so the DNA nanostructures which can be used to build disease targeting units for diagnostic as well as therapeutic purpose. That means, the same nanoparticle which can diagnose as well as which can carry the therapeutic agent, it is called as theranostic nanoparticle, okay. And again this hollow structures can be designed where multiple pharmacological active species can be caged into different compartments. As I told you earlier, in case of early stage of cancer, if you want to release only this kind of drug, only this box can be opened, and the person who is having this advanced stage of cancer, if you want to have 2 different kind of drugs and which can have the very good therapeutic effect in the cancer which is resistant to the drugs, okay. And also we can use this DNA based nanoparticles for controlled release of thrombin or any kind of molecules. So, here you can see here we are going to use a small fragments of DNA pieces, okay, and this DNA is specific for particular protein. In this case it is specific for your thrombin, okay. So, you can use this self assembly process, we can make a X shape DNA tile and this thrombin will go and bind to this DNA, and when you add the cDNA which is specific for this DNA, what happens is the cDNA will go and bind and it will release the thrombin. So, the thrombin can be released from this DNA. So that is why like we can use the DNA based nanoparticle for the control release of thrombin. If you are putting only one cDNA, it will release only one molecule of thrombin, and if you are adding like a 4 cDNA, then it will release all the 4 thrombin. So similarly, we can use the small fragment of DNA as a key to open the DNA based nanoparticles, and it can release the drug according to our need. And we can also make DNA origami nanopores. So here you can see this example. So here it is a DNA origami nanopores. Okay, so that could be useful for detecting various compounds. For example, if you are having a receptor in this nanopores, when you pass the protein or when you pass the sugar or amino acids, so it can easily detect, and it can be used as a kind of good sensor. And again, this DNA nanopores can be lock and open by using this kind of signals like temperature. If you apply temperature, it can be opened, or light or pH. By using this kind of parameters, we can open the pores or we can close the pores, and this is also a very important role in the controlled drug release. And next one is this DNA origami nanopores can also mimic like your potassium and sodium channels. So here you can see the example. So this DNA nanopore, this is the green color one is the DNA nanopore, and this red color is the lipid bilayer. Okay, and it is selectively acting like the potassium or sodium channels. And here this DNA, this green color is the DNA origami nanopores, and it is embedded in this red color lipid bilayer. And here also the blue color square is your anti-cancer drug or any other therapeutic molecules, and this carrier can be useful for controlled delivery or control release of your therapeutical molecule. So the next application is we can use it for bioimaging application. Here you can see here this gold nanoparticle, it is tagged with a recognition sequence, and we can add the reporter sequence. The reporter sequence is tagged with some kind of fluorescent molecule. So it is binding to this gold nanoparticle and form the nano flare. Once it enter the cells, this sequence will target the mRNA. Okay, so when it binds to this mRNA, what happens is this fluorescent signal will be expressed. Okay, so it is releasing this reporter flare, so it is giving a fluorescent signal. So in this example, so this cervivin is one of the anti-apoptotic, that means this kind of protein mostly expressed in the cancer cells. So when you target this kind of proteins, and it could be useful for cancer therapy. Okay, so how do you stop this? We can stop at the protein level, or we can stop at the mRNA level. So from DNA we will get the mRNA, and from here you will get the protein. So in this example, so cervivin, we have to stop the cervivin protein. So we can stop at the mRNA level. Okay, so the sequence which is tagged to gold nanoparticle, so this mRNA will bind to this. Okay, so what happens is when the mRNA binds the sequence with the fluorescent signal, it will be released. So once it is released, it will give the fluorescent signal. So here you can see here the cells which have the more amount of cervivin, you can see here more amount of fluorescent signal when compared to the control cells where the cervivin expression is less or no expression of cervivin. So we can use this DNA based nanoflare for various diagnostic as well as for therapeutic applications. And the next application is we can also use this DNA based nanostructures for energy transfer and photonics. Okay, so here the DNA nanostructures are intrinsically more rigid than the double stranded DNA, so it can be used to build a longer photonic wires. So using this we can harvest light. It can act like a light harvesting complex, okay, by spatially clustered and aligned to create a new generation of photonic wires. So we can use the DNA as a photonic wires, and we can also use it for energy transfer as well as photonics. So let us see some of the drawbacks of DNA origami. So here the DNA origamis are not stable in various conditions. It requires special care. For example, pH has a drastic effect on the structure of DNA nanostructures. In low pH, the DNA may be depurinated, and in high pH, the hydrogen bonding between DNA strands will be disrupted. Okay, and also the heating many chemicals and some organic solvents may denature the double stranded DNA, and again the enzymes like DNase enzymes which can destroy the DNA strands, and it can also destroy the DNA backbone. Okay. So, thus we have to store and handle these samples very carefully, and again the ions present in the solutions have a strong impact on the DNA structures. Okay. So, at low ionic strength the DNA structures will decompose, and at high salt concentration which leads to aggregation of the structures, and also the molecular tensions and mechanical forces also have the negative effects on the structures. So, we are also example for the program self assembly. So, if you see that we are also originated from a simple atoms, the atom combines and forms the cells, and the cells combine and forms the tissues, and tissues and combine and form the organs, and organs combine and form the complete body. So, if you understand the self assembly, we can also make such kind of complex structures and nanoscale devices and everything, but our understanding is of self assembly is still in the elementary level, and as a summary, the self assembly is one of the few practical strategies for making ensembles nanostructures, and here the DNA will be the key player in bottom up nanotechnology. Okay. So, to make more complex DNA structures you need more highly developed computer programs. Okay. And also we are limited with number of software programs to develop to understand the DNA structures or making the designing the complex DNA structures, and each of the program has its own advantages and disadvantages, and additionally we have to sequence optimization is very very important. Okay. And again the DNA nanotechnology and the DNA origami, it has opened a novel pathway for addressing many previously impossible challenges. Okay. So, I will end my lecture here. I thank you all for listening to this lecture. I will see you in another lecture.